Today, I will talk about the penicillins and cephalosporins. Both of these drugs are a cell wall synthesis inhibitor, means they kill the bacteria by stopping the synthesis of cell wall. Both of these drugs are called as the beta-lactam antibiotics because they have beta-lactam ring. The beta-lactam ring is composed of 3 carbon atoms and 1 nitrogen atom. Thiazolidine ring is attached to the beta-lactam ring in penicillin, whereas dihydrothiazine ring is attached with beta-lactam ring in cephalosporins. The R1 side chain, located at 6th position in penicillin, is responsible for bactericidal action. The R1 side chain, located at 7th position in cephalosporin, is also responsible for bactericidal action, whereas the R3 side chain, located at 3rd position in cephalosporin, is responsible for the pharmacokinetic profile. Now, normally, the bacterial cell wall is made up of peptidoglycan. The peptidoglycan is composed of sugar and peptide chain. These are the sugar molecules, and this is the peptide side chain. N-acetylmoramic acid combined with the N-acetylglucosamine. The peptide side chain is normally attached with the N-acetylmoramic acid. Now, transpeptidase enzyme cross-link the two adjacent peptide chains and thus causing rigidity in the cell wall structure. The process is called as the transpeptidation. Normally, the transpeptidase binds or recognizes the D-ALA sequence and thus cross-link the two peptide chain. If you're going to take penicillin and cephalosporin, these both drugs mimic the D-ALA sequence and thus binds with the enzyme transpeptidase and inhibiting its activity, as the activity of transpeptidase is to cross-link the two peptide side chain. So if the two peptide side chains are not cross-linked, there will be inhibition of cell wall synthesis because the cross-linking cause a rigidity in the cell wall, and in this way there is permeability. The inability to cross-link of two peptide chain lead to high permeability and thus the bacterial death. Now, one more thing is that the penicillin are more effective against gram-positive bacteria. The reason is that in gram-positive bacteria, the outermost covering is made up of peptidoglycan, whereas in gram-negative bacteria, there is also presence of lipopolysaccharide. That lipopolysaccharide basically shows resistance to the beta-lactam antibiotics. Beta-lactam antibiotics in gram-negative bacteria passes through porine channel, as lipopolysaccharide also have some proteins and they are responsible for the transport of beta-lactam antibiotics into the bacterial cell. Now we have natural penicillin, penicillin B and penicillin G. Penicillin V is we take penicillin B in as oral and they are responsible for less severe infections, while penicillin G is responsible for more severe infections. Now there are some techniques followed by bacteria in order to um prevent the beta-lactam action, as beta-lactam is responsible to kill the bacteria. So what the bacteria do in order to protect itself, the bacteria secrete beta-lactamases enzymes. Those beta-lactamases enzymes basically kill the or degrade the beta-lactam ring, right? So inactive beta-lactam ring cannot cause bactericidal action. Furthermore, the bacteria causes decreases permeability to drug. So if a drug cannot enter into bacteria, it cannot perform its function. Moreover, uh, there are efflux pumps in the bacteria, and they thus, uh, if an antibiotic enters the bacteria, they normally uh, prevent the entrance of antibacteria into the bacteria, and they normally excrete or remove that, uh, or you may say expel out the antibiotic outside the bacteria. So an efflux pump is responsible to expel the antibiotic out of the bacteria, and in this way bacteria prevent itself from its destruction. Now the other is that the bacteria alter its enzyme, protein binding penicillin. So if the there is alteration in the enzyme, the beta lactam antibiotics cannot bind to that enzyme. So if a beta lactam antibiotic cannot bind to the enzyme, then it cannot show its effect. So this was all about in today's lecture.